Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. We are taking a break this month. While we do so, we invite you to revisit our previous podcast episodes. Today, or this week, our throwback episode is episode 59 with Dr. Nicola Smith. I recorded this episode very early in the morning in October 2021 from our brand new home office. We've upgraded from recording in my car and my tiny walk-in closet to finally a very comfortable home office. Nicola's story is extraordinary. In 2019, she and her team realized that the construct containing GPR37L1, or GPCR of interest, was not only in the wrong vector, but was inserted backwards. And this construct had been used in at least 2 high-impact publications. Nicola was faced with the hardest decision of her career, which was to report the error and face the consequences. Hear more about her story and how she dealt with this difficult time in her career and life. During our conversation, Nicola also shared with me how she and her team are now working to avoid replicating such errors in their lab. Before we revisit this episode, we are excited to share that the Dr. GPCR Ecosystem 2.0 platform is now open. Although we plan on opening the ecosystem slowly to resolve any kinks as soon as possible, we invite you to visit us at ecosystem.drgpcr.com, register as a site member, for which we will approve you, and explore the new site. As you know, access is restricted to members of the GPCR field, and each sign-up will be approved based on the involvement in the field. When you sign up and you get pre-approved, you'll be able to enter the ecosystem. You'll also have the option to select a plan and get access to all things Dr. GPCR, including the upcoming Dr. GPCR Summit in October, access brand new podcast episodes before they get released to the general public, access our new group discussion and forums, the job board, our learning center, where you'll be able to take or create and share a course, and also discover GPCR companies and much more. Take advantage of everything that the new GPCR dedicated online playground has to offer today by visiting ecosystem.drgpcr.com to start your GPCR journey. This is the only place where GPCR scientists, trainees, and GPCR organizations can thrive, and where it's only about science and GPCRs. Think ResearchGate meets LinkedIn meets Amazon. Also, make sure that you mark your calendar for the 3rd edition of the Dr. GPCR Summit. This year, the summit will be held between October 10th and 16th. Stay tuned as we are working on the 2022 Summit program. Visit drgpcr.com to find out more about all of our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and I have the pleasure today of having with me Dr. Nicola Smith. Hi, Nicola. How are you?
Hello. I'm good.
I'm so glad we're doing this.
Oh, me too. I love the podcast. I think it's a great initiative, and just to hear kind of casual conversations with our research heroes is wonderful.
Thank you. So I know we talked about this before I hit record, but what time is it where you are?
Oh, it's 7:15 in the morning. So
and it's 4:15.
Yeah.
Yes, 4 in the afternoon here in the Boston area. We were just discussing before we hit record that last time you and I gave a talk in the same session, it was 4:30 in the morning for you.
In the morning, yeah, that was very, very early. But this is very reasonable, and I'm fine with that. I'm just worried about a 2-year-old coming and joining because he hasn't woken up. Uncharacteristically, he has slept in, and his room is right next to my study, so he may join in.
Well, it would just add to the beautiful diversity of the podcast. We haven't had a 2-year-old yet, although my 6-year-old loves it when I make podcasts, and I was thinking of maybe asking him to say, welcome to the Dr. GPCR podcast and edit it in. But I wasn't sure how professional that would look. So
definitely I'd be fine with it.
I would be fun. I think it would be awesome too, but I haven't made that decision yet, although he loves the podcast too. All right. So let's jump into it. Can you walk us through who you are as a scientist, Where were you before opening your own lab? What got you into the GPCR field?
I guess so. I'm a molecular pharmacologist, and I have been mad about GPCRs since my undergraduate studies. And I went on to do honours and a PhD, all in GPCRs, and then got a prestigious Australian fellowship, which has been instrumental in most Australian GPCR researchers' careers, to go overseas. And I went and worked with Graham Milligan in Glasgow for 4 years as a postdoc. And then one of the nice things about the fellowships is that they give you 2 years upon return to Australia. So I returned to a cardiovascular laboratory that also worked on GPCRs at the Victor Chang Cardiac Research Institute. So my idea was go overseas and really learn all about the molecular studies for GPCRs, and then come back to Australia and learn animal models so that I could understand them better. So that's kind of all my friends, even those who don't have a science background, know that I'm obsessed with GPCRs.
As a child, as a teenager, were you what was the science theme that you're interested in? I've interviewed people who said, oh, physics or mathematics or biology. What was it in your case?
For me, I came into I guess science belatedly. Both of my parents are journalists, so, and they, in terms of support for maths and sciences, that ended in primary school. They were like, I'm tapped out. I can help you with the wording, but nothing beyond that. So I was always interested in journalism, and my dad's a sports journalist, or was a sports journalist, and in particular, that's what I wanted to do. So I started university wanting to do sports psychology. My school kind of pushed you into things that you were good at, and I was really good at science. So they just kind of funneled me through that, and I loved it, but I didn't have a particular passion. I knew I didn't love physics, but I did fine at physics, but I knew I didn't love it. But chemistry was cool. And I think because I was forced through a very defined course at my high school, and I wasn't able to do anything to do with history and the subjects that I've always been fascinated by. I had the privilege at university of doing an arts degree and a science degree. So in my science degree, I was going to major in psychology, and in my arts degree, I was going to just follow my passion. So I did all sorts of history subjects and history of philosophy of science, which I loved. I absolutely loved it. And at my university, Melbourne University, they also offered a history of Australian sporting culture. So I loved that as well, because it told you the whole history of Australia as a nation since colonisation, and how sport played a role in integrating all the different communities. So I had a ball, and then suddenly I took pharmacology. And, in Australia, you don't offer pharmacology until the second year course, and I was doing a double degree, so it wasn't until my third year that I took pharmacology. And it changed everything. I had to put all of my psychology subjects into my arts degree, and I did a double major in pharmacology. And from that moment on, I was absolutely hooked. I had to do pharmacology. So, and the only reason I took pharmacology was because of the maths. Sorry, Yamina. Because of my maths assignment in year 12, which ironically actually was about modelling the drug distribution. So I just had to model paracetamol as an example of its pharmacokinetics. And I went and spoke to the local chemist and talked about dosing and getting a maintenance dose and a loading dose. And I had to just model that mathematically. But that planted the seed that meant that even though I was doing neuropsychology, I just wanted to do that pharmacology course. And I didn't get in the first year because I didn't have the prerequisites. The second time I applied, I got in. So it was that one math subject was so instrumental. And ironically, I hate pharmacokinetics, but that's what got me into it.
It's just so interesting. I don't think I've heard of anyone I interviewed so far who wanted to go into journalism and sports journalism, which is it's unique. I mean, on the other hand, the skills that you've developed while thinking about that course and and that career path is helping you because you're using your ability to express yourself and to communicate information, but to a whole different audience.
Yes, definitely. I think that, if people would look at my strengths, they would say that my strengths were synthesizing information and writing and communicating. I'm not the kind of person that's going to be able to develop an assay from scratch and get into the buffers and know exactly what each buffer is doing. I need to employ or surround myself with people who are good at that, because that's not my strength. My strength is looking at the literature and putting things together and communicating that well.
Lovely. And then you mentioned that there was this course where you first heard about GPCRs and that got you hooked. Was it the pharmacology course?
Yes. And initially, I guess in biochemistry, we heard about we learned about the small G proteins.
Yeah.
And they already kind of captured my interest, but it was pharmacology. And can I just tell you, my undergraduate pharmacology, I was taught by Arthur Christopoulos.
Why am I not surprised?
So, and he was up and coming at that stage, very, very ambitious and so enthusiastic. And he just set up his own lab in Australia, having returned from a really incredibly successful postdoc overseas. And back then, he was a pharmacist. He is a pharmacist by training. And he just followed his passion for science. So he came into the classroom and that just oozed out of him. Anyone who's seen him give a plenary knows that he's a fabulous communicator, and you can't help but walk out excited about allosterism. He was teaching me about GPCRs for the first time. And another really important teacher that I had was Michael Liu, who's a real purist. So he's written a lot of kind of reviews about receptor theory. And he spoke to my interest. It was really about how things work and how drugs work, and it just was awesome. And within our department, I presume it's universal, but everyone worshipped Terry Kanaken.
But I think everyone still worships.
I should hope so. I should hope so, right?
I'm pretty sure. I'm one of them.
Yeah, oh God, yeah. I remember dragging him to a poster of mine, actually in Il Ciocco in Italy. We were talking about Il Ciocco earlier, and how that's a wonderful place for GPCR conferences to be held. And I remember just being so nervous. And he, of course, is the most wonderful man as well. And he's looking at this fairly pedestrian GPCR research at the time, because it wasn't a fully formed story, and it was just a PhD student. And he gave me the time of day, and he gave me that time looking through it, and I completely fangirled on him. I was just like, "Oh, Terry!" Terry's looking at my poster. Yep. But I was in a really strong pharmacology department and being taught by one of the best GPCR researchers that I think we've seen. So it was pretty,
as they say in French, the crème de la crème.
Oh, yes, exactly.
It's funny that you mentioned, dragging Terry to your poster. I had a similar encounter with Terry. I think I was a PhD student at the time, and it was a GPCR retreat. And we were, I believe, in London, Ontario. And he happened to stop at my poster, and I was talking about my story, and I was ecstatic. But on the other side, I was seeing my boss, who was looking at me saying, "Hey, Terry had stopped at your poster. How fun is this?" And he was fantastic. I mean, the interest, the attention that work got he was there 100%. I love talking to Terry.
Yeah, he's so he just has an ability to, because you do sometimes go to your heroes and drag them to your poster, and it takes a lot of guts and you feel ill doing it. And some of them aren't as good at giving you that time. But Terry, like you said, he was in the moment and paid attention to it as if it was like a cell paper, even though it was clearly not that. And I'll never forget that, because that was such an important moment in my research, to have your heroes actually turn out to be like you had hoped your hero would be.
Exactly. So I asked this from every guest. Any favorite GPCRs? Do you have a favorite GPCR?
I don't. I started off, I actually did honours with Arthur and with Patrick Sexton, and that was with the serotonin 5-HT2C receptor, and I loved that receptor. But then I moved to the angiotensin receptor, which has so many tools, and it's great to study. So I thought I'd always be an angiotensin person, but then I've ended up working on orphan GPCRs, and I think I actually just love that whole category of orphans. I love how hard they are. The number of people who've told me that I made my career path so impossible by choosing orphan receptors to work on, and they're all absolutely correct. But I just love that untapped potential, the fact that their story hasn't been told yet, and how can I get information out of this damned thing without actually having a hook. And one of the ones we're working on at the moment, I'm absolutely fascinated by, GPR 161, and I've got a graduate student working on that, and it's found almost exclusively in the primary cilia, and so each cell has one primary cilia to kind of sense what's happening in the world. And GPR 161 sits just next to the cilia or is recruited into the cilia, and it acts as an ACAT itself, so PKA anchoring protein. And the regulatory subunit actually directly binds to the C terminus and the tail, but also PKA phosphorylates the receptor as well. So it's a totally different way of us thinking about kind of the canonical GPCR pathway. It's a different way of the GPCRs acting, and that it's thought that perhaps that's through a constitutive activity because it's all happening in the C terminus and through phosphorylation, and it's more subcellular localization that's driving its function. So we're trying to understand a bit more about how the receptor itself is functioning. But this it's also, when you follow it down the rabbit hole, it's taken me into a whole new world of science that I didn't know. My poor graduate student, I'm constantly saying, what's the latest? And she's writing a review at the moment, and I keep pushing her for the drafts, not because we need to meet a deadline, but because I'm dying to read her synthesis of everything, because I am able to keep an eye on the literature, and I have a much more of a surface level understanding compared to where she's at. So it's such a cool receptor.
I think it's a fantastic and cool story, and it reminds me of Ben Myers's work with the smoothened receptor.
Oh yes, he's fabulous. So
very very similar. Where smoothened is expressed also in the cilia, and it also binds the active site of PKA.
Yes. So he's done quite a bit of work around this area, and his work is beautiful. It's really
I think
nice.
so too. I think so too. We're gonna by the time we release this episode that we're recording, we would have already released the episode that I recorded with Ben. But fantastic story. Yes. Here, let me take a step. Let's take a step back. So we said serotonin, angiotensin, and then orphan receptors. What happened between angiotensin and orphan receptors? How did you get interested?
Oh well, I blame Graham Milliken. He's Graham is when I came to join his lab, he was working on kind of recently de-orphanized receptors. And so I guess I went from angiotensin to, in the Milligan lab, they were kind of working on things with very little tools, but at least they had some tools. So I started working on the fatty acid receptors when I got there, and they weren't even the free fatty acid receptors. They were GPR 40, 41, and 43. And we actually wrote the IUFAR nomenclature review where they became FFA 1, 2, and 3. And that's where I really got into it. And I thought, take one step back. Orphans altogether are very cool. And what can we do when we have no tools, and how can we understand them? So it was that 4 years in his lab that gave me a lot of the understanding and the tools myself to know how to approach it in quite it's pharmacologically the stuff that I do compared to, say, Brian Roth, who has an amazing program on orphan receptors. And I was always interested in orphans, and in the time that I've been working on them, some of the ideas that I've had, he's done them, and he's done them way better than I would ever be able to do it. But it's quite rudimentary pharmacology in many ways, trying to get a handle on orphans. So I learnt a lot of that with Graham, and then I came back to Australia with an orphan in mind that I wanted to work on, GPR 37L1, because it had been implicated in blood pressure. And since my PhD in angiotensin, all of my work has been within the cardiovascular space. So, yeah, that's how I made that jump. It was kind of like it was a stepping stone into the unknown with the recently de-orphanized and low affinity. That low affinity GPCRs.
Totally. I always like to know what the behind the scenes stories of how people jump from one family of receptors or one family of G proteins to that. So you mentioned that you're really interested in orphan GPCRs where there aren't a lot of tools. That takes me to my next question. What type of tools that you have found to be transposable from one orphan to the other? I'm thinking here, there is a bunch of orphan GPCRs, and you have most likely built a platform, kind of a plug and play, into switching out the receptors and studying different receptors with those rudimentary tools. How does that look like?
So the first thing we do with every receptor is put it into flip-in cells, so inducible flip-in hex. And the reason we do that is because anything we find is going to have most likely low potency or off-target effects. So having that inducible cell line means that we have an automatic control every single time with non-expressing and then expressing. The other thing we've done is use quite downstream reporters. So the CRE luciferase, SRE luciferase, NFAT, so that we have a general idea of what the G protein might be that the receptor is signaling through, and we're relying on constitutive activity. And by having it so far downstream, you get an amplification somehow of what you're looking at. That, of course, presents its own problems, and I think we'll probably touch on that a bit later, that if you're relying on something so far downstream, you're missing a big part of the picture, and things can be coming from different pathways that you didn't realise. So, and then we've recently started using the beautiful nanobret work from ASCA in a way, and we've also got the TruePath. We're having a bit of trouble getting that to really sing for us in comparison with the nanobret assay.
Interesting. You mentioned GPR 161. Is it a receptor that? So what does it do? Think of it. I've never heard of GPR 161. Why study GPR 161?
Well, we study three orphans at the moment in the lab. We study GPR 161, GPR 146, and GPR 3701, which is the one that I have worked on for 10 years. So 161 is a bit of an enigma. It's definitely involved in development. Knockout mice, half of them embryonic lethal, but the other half actually survive, but aren't well. So there's already that kind of conundrum of why half escape that kind of developmental block. The reason I first became interested in it, I noted it a number of years ago before any of this understanding of the cilia, was because in zebrafish there was a heart looping phenotype. So that's what I was interested in, the fact that there was a cardiac developmental role. But that doesn't seem to have held. I've never seen that. That seems to be specific to zebrafish. And we know that, with morpholinos that you can get some aberrant or inaccurate phenotypes. So really it seems to be developmental sonic hedgehog signaling pathway roles.
And actually that I was listening to what you were mentioning, and the question escaped me. It was so fascinating what you were saying. Goodness. I was listening at the same time. I was, I have to ask you this question, and you think I remember what it was. All right, let's move on to the other GPCRs. So I understand that you're working on orphan GPCRs, but do you? Oh yes, I remember now.
146.
Yes, 146. But before we go there, you mentioned that you really had training in hardcore pharmacology, and then you pursued training in animal models.
Yes.
Does your work take you from the hardcore pharmacology to the animal models?
Yeah, so currently the composition of my lab takes me from computational work, which I am not an expert in, and I get all of my advice and co-supervision of my student with Irina Kufareva at UCSD. She's brilliant. She's such a brilliant mind and such a wonderful computational chemist and computational biologist. And she's done a lot of work around structural stuff as well with Tracy Handel and others. So she helps us with our GPCR coin pocket methodology that she co-developed. And so our idea is that we kind of identify ligands for orphans through non-specific activity and then transfer that into our in vitro assays. But we also at the same time have animal models using the knockouts. So we've only been working with GPR 3701 knockout mice, and we've had a couple of different lines that we've worked with. And we've done all sorts of different things with those, just looking at endpoint blood pressure measurements. We've done microtelemetry, we've done metabolic studies, and they've been fantastic. And this receptor is a nightmare to work with, particularly the animal models. It's really, to misuse a genetic term, it's kind of like it's not fully penetrant in that, and that's not using it in the genetic sense at all, but we get every single study we do, we get a phenotype, but it's not consistent. So it's not like if you think it's a sympathetic, so we've shown that there's gender differences or sex differences. And we did really, my graduate student, Meg Mowat, she's phenomenal. And she's done this beautiful radiotelemetry studies and shown a sympathetic role. But the measures you would expect to indicate a sympathetic role, it's like not significant, not significant, not significant. And then one measure or two measures will suggest there's a sympathetic role. And then the same with the blood pressure regulation in endpoint stuff. And the same with the metabolic phenotype. There's certain elements that are clearly affected by lack of receptor, but it's not a consistent. So it's not like you go through the glucose tolerance, the insulin tolerance, then the weight difference. So you never get, it's not like a really overwhelming phenotype. And that's been terribly difficult. It's been very difficult to understand, very difficult to publish. And ultimately, I guess it's led me to, I think this frustrates my students who are working on it. I've started saying, I just don't think it's not a reliable or reasonable target. And we've spent 10 years on it, but it's not it may well be a target for something else, a neurodevelopmental or neuroprotective target, but it's not a cardiovascular target.
Wow. But you had to do the work in order to get to that conclusion, which is, I'm sorry, but it sucks, but still.
Oh, yeah. I feel like my whole career has been based on showing that existing studies are either incorrect or overblown. So the 3701 story, I got involved in it because I found a paper in BBRC, and it was a pretty flawed paper, but it showed that if you knocked out GPR 3701 in mice, they had a 62 millimeter mercury increase in blood pressure. So basically, that's bonkers. So enos, endothelin-1, all of those things that are contributing to blood pressure, you only see, say, 15 to 30 millimeter mercury changes in blood pressure. This was 60. So I didn't think it was going to be 60 when I did it myself, but I thought even if that wasn't a very well controlled experiment, it should be 30.
I would have guessed 30 too.
Yeah, right. And in fact, I took the paper that when I found it to Graham Milligan, and I said, look at this, we should work on this. And he saw the GPR 35, which he was already working on. And he's made huge advances on GPR 35. And he said, well, he was interested in that rather than 3701. And I needed a way to establish myself and my independence from Graham. So I said, all right, well, I'll do 3701 because that looks pretty cool too. That's the one with the really big phenotype. They made mice and tested it, and they got the same crazy results as us, really marginal, inconsistent. So we've both been hurt by that original paper. And we just had a paper come out in American Journal of Physiology just, I think, maybe last week or the week before, where we tried to reconcile some of the differences between the original paper and our paper. And I'm actually really proud of that. It's a small paper and it's not in a big journal, but I'm really proud of that study because we went to the effort of, rather than just leaving it, of actually really detailing, this is how we did the experiment. We got their mice. This is how we did did the same experiment that they said that they did using their mice, and we did not get the same results. And we think this is why. And I think it's important, if it stops anyone from doing what I did and went 62 millimeters of mercury, there must be something there, and pursuing that, then I think that we've done a greater service to science than if we discovered something really hot and got a Nature paper from it, say, and it not having traction. I think that this is something that's really our way to contribute to science, is publishing these papers. And I've got a few of them now where I've, from my PhD as well, where it's like, this really sexy mechanism is not true. So less sexy, but really I think it's very important.
I think so too. I think it's very important to, do the work properly, have the right controls, and then publish the data, no matter what the data says. And it's funny because as a master's and PhD student, I'm sure you know this, when you're working in the lab tirelessly, and at some point you only get negative data. And we were joking, and we were like, maybe we should, put together the Journal of Negative Data. Yeah. So that it saves people time saying, we've done this is what you've done, and it's not working. So, you could try, but this is what we've done.
Yeah. Yeah. And I think that one of the really good things, and with shouldn't use the word revolution, but it feels like it's a real reimagining of how we publish these days. And I think that there's that push for, I love the fact that all the journals now are getting you to put bar graphs that have the data points on them. Nothing is hidden behind this column and a standard error of the mean, which that you can have 4 or 5 different permutations to give that same result. And if you'd seen the original data, you would, in some cases, you'd go, that's so obviously wonderful. And in others, you'd like, are you serious?
Yeah.
That's a kind of a coin toss, really. And it's significant because you got a really good outlier that dragged your results up or down. So I love that we are having that transparency, but also I'm loving the fact that, well, I don't think it's rewarded in our grant funding. The scientific community is encouraging and embracing the idea that we put out our negative results as well as our positive. And AJP, when I wrote to them, I wrote the cover letter saying, this isn't, we don't have all the answers, but we believe this is a really important reproducibility study. And they were really very keen to publish it for that reason. They believe very strongly that these kind of studies needed to be out there. And I think it's important that we change the course of how we're publishing for those reasons.
I agree totally. So this takes me to my next question, which wasn't in the episode outline. And you just gave a talk when you were up at 4:30 in the morning about what happened when you realized that there were sequence issues with your DNA and you had to retract. And you're right, I don't like the word retract either, but you had to course correct. Let's call it course correcting your publication.
A very public correction, course correction. Yeah, look, I have had two retractions, and they are the two papers that were most significant in my career as an independent researcher. They set me up for a fellowship. They set me up for, I just started my own lab, and my entire research program was built on the work of those two papers. And while I was on maternity leave, I had an honours student start in the lab, and he just couldn't get the cloning to work. And it's risky to put an honours student in Australia. We have an honours program that is, I guess it's like masters overseas. So we actually, some people do masters in Australia, but that's, I guess, viewed as a lower, even though it's a higher qualification, it's not rated in the same way as our honours programs. And in Australia, you go straight from honours to a PhD, unless you didn't get good enough marks, you do a master's and then go into a PhD. It's all completely the wrong way around, but that's how we do it. So our honours students have about 6 months in the lab. It's a 9-month degree or course, and so it's very risky putting them on a mutagenesis project, because mutagenesis is in the hands of the gods. And he just couldn't get the mutagenesis to work. And when I came back from maternity leave, I just said, this poor guy's been working his butt off trying to get it to work. Let's just order it in from GenScript. And if we're going to order in all the mutants, we should just order in their wild type as well, so that it's all consistent, even though the sequence is identical. And we already were using pcDNA3. We got them to put it in pcDNA3. And he got it. And I always make my guys run maxi preps on a gel to make sure they've got nice, good quality maxi preps, and that it runs at the right size. And he ran his old wild type receptor on the same gel, which is great, good quality control. Completely different sizes. They ran at completely different sizes, and they were both supposed to be pcDNA3, GPR3701, untagged. And we were puzzled, obviously, and did a lot of investigating, and realised that what we thought was GPR3701 in pcDNA3 was actually GPR3701, but inserted backwards into a yeast vector. And the reason why we didn't pick it up is because it was a single restriction digest, so with Hindi3, and directionally you need T7 to go into the start codon and into GPR3701. The yeast vector has a T7 at the 3' end, so the sequencing read from T7 into the start of the receptor, but because it was the yeast vector, it was backwards. And the was a volunteer in the lab who made that construct, and just a series of circumstances that on their own wouldn't have been a problem, but just kind of a comedy of errors, I guess, and circumstances meant that we didn't pick it up. So it was a construct I'd said to her on her last week of working in the lab or volunteering in the lab. Can you just do that one while you're doing making some yeast stuff for me? Just do that one as well, because that might be handy in the future. And so she did. And she checked it for T7 sequencing, but the 3' sequencing didn't work. So she only had that to go off, and she didn't look as closely, and I can certainly forgive her for that. She didn't notice 2 base pairs right at the start of the sequence that were different, because it's easy to put that down to that first part of the sequence can be,
of course,
a bit hit or miss, right? But she confirmed that T7 went into the start code on the COSAC, start code on GPR 3701. That was then maxi prepped and just put in the freezer. And I had a graduate student come along, Tony No, who's been very severely impacted by these retractions. We had yeast data showing the receptor was constitutively active, and he was working with a tagged version of the receptor in human cells and didn't get the same result as we were seeing in the yeast. And so he tried the untagged version of the receptor. He went to the freezer, grabbed that version, tried it, and it confirmed the constitutive activity that we'd seen in yeast. So you'd never expect a dud construct to give you, first of all, have activity rather than no activity, and second of all, confirm results you'd seen in yeast. And he didn't sequence it because it was in my -80 collection, and he didn't realise, none of us realised that there was a problem and that it hadn't actually had the sequencing work for the 3'. And those 2 base pairs that were slightly different indicated a difference in the multi-cloning site. So when this honours student found the size difference, we did the sequencing, and I noticed those 2 base pairs and was able to work out that the multiple cloning site was from a different vector. And my postdoc at the time, James Coleman, who also has been impacted by it, he was supervising the honours student. He worked out he did a whole lot of detective steps and was able to work out that it was in the yeast vector, and together we worked out it was in backwards. 6 months of experiments then to try and work out the implications for our research program, and the implications were that it was wrong. We couldn't reproduce, if we used the correct sequence, we couldn't reproduce that constitutive signalling we'd seen in yeast. So we contacted the journals, Science Signalling. They were fantastic and immediate. They surprised me. They didn't necessarily know the right steps. They didn't have a clear process, so they had to kind of go away and work out how to deal with it. But open communication, really fast, fantastic, came back, yeah, you're definitely going to need to retract it. And they did it quite quickly. Nature Chem Biology were very slow, but I don't think it was their fault. We contacted them and said, would you please consider a correction rather than a retraction? And the reason we asked them for a correction was our Nature Chem Biology paper was a computational and wet lab paper. It was on a methodology we called GPCR coin pocket. And basically we said, we know nothing about orphan receptors. What can we learn from liganded receptors? And some beautiful work by Brian Shoykert showed that if you looked at from the perspective of ligands, you could actually rearrange, compared to the phylogenetic tree, you could do a chemogenomic tree, and you'd find receptors that ordinarily you wouldn't think of being closely related were chemogenetically. And so you could use ligands from those adjacent receptors. But orphans are not represented, because in that, obviously, they don't have any ligands. So we said, well, obviously there's something about that three-dimensional shape of the pocket and the identity of the residues within the key points within that orthosteric binding site that shapes the type of ligands that the receptors respond to. So let's see what our orphan receptor pocket looks like in comparison to liganded receptors. And then we would pickpocket, was the idea. We'd pickpocket the ligands from those receptors and try them on ours. So the method itself has been independently validated by other groups who not only have used it for GPCRs, they've actually used it for other proteins. So it's actually a fantastic method, and that's all work that was done with Irina Kufareva and originally also with Ruben Abigan. The wet lab stuff was the validation of the ligands, and it was all based on using that incorrect receptor construct. So our argument to Nature Chem Biology was the methodology is right, and we didn't think that the paper warranted retraction because it works, even though our wet lab validation doesn't now stack up. Other people's does, so that should hopefully please be enough. And they said, okay, we appreciate your request and we'll send it back to the original reviewers. And I think they had one reviewer that just took months in each cycle, from my understanding. So while they were trying very hard to push it through, because the science signalling retraction was already out, so they would have known that people would be drawing kind of links between that and the Nature Chem Biology paper. But it took ages. They came back and said, no, they gave us another opportunity. They said, if you can provide us with any other validation that this works with another orphan, we will consider it as, we'll review it again for the quality of the science, but we'll consider a correction in that case. And we just said, look, that's months and months of work. We just simply can't. We're not set up with any other orphans at the time to do it. So we had to retract it. But they, again, I thought were very reasonable and gave us an opportunity to publish a Matters Arising, which they, again, it was uncharted territory for them, because they said usually we have Matters Arising in response to someone complaining about a paper. Yeah. And so they have this is all wrong, blah, blah, blah. And then the authors of the original paper come back with, no, we think we were right all along. But this was us saying, hey, guys, there's a big problem with this paper, and this is why, and this is what we think it means. So they, it was a bit different for them to try and deal with. But again, it was incredibly, incredibly long and protracted process. So, and definitely the most stressful period of my life, scientifically or otherwise. It affected my collaborations, people who were innocent bystanders. Irina Kufareva was co-corresponding author on that Nature Chem Biology paper, and she's published some stunning papers in much higher journals, but still it was really important as she was herself establishing her independence. So she got caught up in it all, and it was horrible. And other collaborators who did other things on the science signalling study, who it meant, they lost those papers, and the graduate students who worked on it, that was their main kind of stellar papers as well, and that their careers are at that point where that was all they had, and suddenly that was taken away from them. The stress on the students in the lab was insane. And then just the personal stress of just coming back from maternity leave. We were also renovating our house, so I was living with my in-laws who had no appreciation or a sympathy for someone who was being contacted well into the night during dinner. That caused a big drama. During dinner, going through an investigation. And so I was constantly just attached to my phone and preoccupied and not present with my family. and it was really tough. I remember he's now 5, but at the time he was, I don't know, 3. He started telling me, "Oh, no talking, mummy, I'm working, and I've just got to do this, just got to send this email," and things like that, that were not okay with me, and really hurt to see that that was their experience of their mum, was that she was saying, just wait, I have to do this. And that work was just so present in every facet of my life. And it wasn't in a good way. You know, when you're on something hot, you just want to keep going, right? And
yep.
When the kids go to bed, I'm still sitting at the computer, even though I'm a morning person, not a nighttime person. And this was different. This was during meals. This was while I was feeding my 9-month-old. It was just, all consuming, really horrible.
I can't even imagine, but to kudos to you. Honestly, I think I saw your presentation and a longer version of the timeline of the events, and I could feel it came through that it was a very difficult period for you, for your lab, for your collaborators. But I still think, and I'm pretty sure, well, I know because you've shared also the tweets, it was the right decision. Yeah. And it was kudos, basically.
I'm still overwhelmed by the reaction of my peers. And it's for several reasons as well, right? But first of all, yes, I had to do it. I had to, morally, I had to do it. But I just assumed that was the end of my career. I thought that even my funding agency was going to withdraw my funding because it was all based on these papers. They didn't care. They were happy that I was doing the right thing and that I'd told them. They just said, thanks for letting us know. They've been great. I just thought, well, I'll never get. I actually withdrew from a conference. I stopped putting in grants. I stopped. I didn't put abstracts in for a conference. I didn't do anything. I just didn't want to be seen. I couldn't face my peers, even though it wasn't fraud. It clearly wasn't fraud. I just felt the shame of having these retractions so intensely, and so I've been humbled and overwhelmed, and there's just so much emotion from people who still contact me to just say, oh, I've just heard about your story. That's amazing. And I guess I'm saddened by the fact that almost universally the reaction is, God, I'm always in fear that that's me. And the fear is multi-layered. It's that they don't want to have something out there that's wrong. They don't want other people trying to follow up work and wasting their time based on something that's wrong. But also their fear is the ramifications of having a retraction and what the word retraction means, and that it is a smear on your track record. And I think that's a real shame, and I think that's where we as a community need to work together to make sure, not because, obviously, career-wise, I could do with some help, but not because of purely selfish motives. I think that we started off this podcast by talking about how many papers that we've all published that are this really exciting kind of idea or hypothesis that people have been now saying is the new way and the new mechanism of something acting. This isn't real, it's not right. And that's not because we're trying to correct fraud or whatever in every case, and nor are we necessarily trying to correct something that was as stupid as a receptor backwards in a yeast vector. Sometimes it's just because of lab conditions, the handler of the animals, maybe there was an infection in the animal house that we didn't realise, or our own work with the animal model for GPR 3701 shows you that even in the same lab with the same people, you can get variable results that you can interpret differently. And I think the fact that we need people to publish that stuff. We really, really need them to be comfortable publishing it and making sure that we're not wasting anyone's time. You hear about graduate students who are put on projects and it's trying to follow up a lead that someone's published, and it ruins their whole, like they could be the most wonderful students coming through their undergraduate, and they've been put on this really cool project and it goes nowhere. And the lack of funding in our environment at the moment, if they don't have the papers and that career trajectory, they might not be able to take the next step and be lost to our research environment, even though they're fantastic. And if we'd actually known there was a problem and come forward. The other thing that gets me is that from my own experience and from talking to people about this, and you know it from even at conferences, right? I remember Paul Insel came up to me at one conference and said, if 10% of that work is correct, then we're lucky, And he's right. There's so many cases where we know that people just suddenly stop talking about a key finding. That they're no longer presenting that at conferences. Why is that? Or they switch away from following up what we would think is an interesting lead. That the paper itself becomes the end of the story when you can see so much more potential. And it might be because they found something hotter. It also might be because they're having trouble reproducing it themselves.
Yep.
Why don't we encourage and reward that in science? That's my
great. That's a great
crusade.
Question. Yeah. I think that's a great question. And you were just mentioning, changing people's career trajectories because they're put on a project that goes nowhere. And funny story or sad story, I don't know. I had a colleague in my PhD lab who spent an entire summer trying to express some transmembrane protein. It wasn't a GPCR, and kept making DNA.
That was their first mistake, wasn't it? They weren't working on a GPCR.
I know. But it turned out that after 3 months, they'd realized that the construct was missing the transmembrane domain part. And so whatever they were expressing ended up in the media, which they were discarding to collect
every
the
single
cells.
time.
Yes. And then at the time, truth to be told, right, nowadays, sending something to sequencing is not only inexpensive, but it's very quick as well. And then 10, 15, 20 years ago, it wasn't the case.
Yeah.
And, no one thought about sending it to sequencing the minute it didn't work the second time.
Yeah.
And I've learned that the hard way too, where I was trying to do something, and after this incident in the lab, everything I touched got sent to sequencing. Amplify, no digestion, no sequencing.
Yeah.
With all the possible primers to make sure that whatever you have makes sense. But, this person spent 3 months and ended up, teaching science at a high school because I think they got to a point where they were like, well, this is not for me. It's not working. I worked 3 months with something that didn't make sense.
Yeah. One of the criticisms I get, on and certainly on the Retraction Watch article, there was one particular person that just kept going for me, saying, Research 101, sequence everything. And the thing is, I learnt that in my PhD as well, because I was sent a reporter construct that was nonsense, and the same thing. Tried to get it to work, it didn't work, sent it for sequencing, it was rubbish. And just that, I guess, as a naive graduate student going, what? People can send stuff that's wrong? It was really eye-opening, but it meant that I always sent stuff for sequencing. And we did send this bloody construct for sequencing. Yeah.
And
it just fell through the cracks.
Yeah.
I had trained the person appropriately and they didn't fully apply my training, but I didn't check it. And they were too junior for me not to be checking it. And so that, there's all those errors that have crept in. And then my graduate student should have sent it for sequencing himself because he didn't know its provenance, but he didn't send it for sequencing. And then every time the SP6 primer didn't work, we always assumed it was because we were having problems with the SP6 primer, but we weren't having problems with the SP6 primer. There was no primer site. So, all of those assumptions going through. But I do get criticised quite often with people going, didn't you know you should always sequence something? And I'm like, I did.
But I think you could have played the lottery and you could have, those two. The way you were describing what happened with that plasmid and what was going on is just incredible. The chances of that being backwards. Yeah. And it's just, I mean, for it to validate, yes,
a gain of function that we had observed in a different assay, for it to validate that function, that's the crazy thing. Yeah. Why does this vector cause a Cre-luciferase to be elevated? And I guess I was saying earlier, the problem with using the wonder of using those really downstream reporters is that they're accumulation assays, so you get a bigger signal.
Yeah.
But the disadvantage is you have no visual on what part of that signaling pathway is contributing to your signal at the end. God knows which part of the signaling of that pathway a reverse inserted yeast vector is triggering, but something's being tickled.
Yeah, I feel like, when these things happen in life in general, not only in science and the lab, these are lessons learned. This had to happen. And it, as painful as it was, as painful as, it still is. I think looking to the future, if you can comment a little bit about what are the lessons learned with this whole situation that happened?
Lots of lessons and lots of reflection and self-reflection. I think now that I'm 2 years through it, I think, because right now, actually 2 years ago right now, 19th of October is when we're recording this, it was the worst, most intense period. We were drafting the letters to go to the journals to tell them. And I look at that now 2 years on, and I think that probably this has been a good thing for me. And that's because it's changed the way I view science. It has made me, it's probably not changed. The changes probably haven't helped my career because I'm no longer motivated by getting that big paper that's going to get me the big, sexy story that's going to set up my grants for the future. I just don't care about that anymore. And that probably will be the end of my my longevity. But all I care about is making sure that we're doing the most accurate science that we can do and that we don't publish something that's wrong. If we don't have all the answers, say we don't have all the answers in the paper. Don't try and spin it. And everyone's spinning stuff these days. And, I come from a journalistic background. I know what a good pitch looks like. But I just don't want to be that scientist. I want to put out stuff that people go, oh no, Nicola's lab, we know she does everything really, really thoroughly because of her experience. I've put out in a lot of processes in the lab, which can be quite intimidating, I suspect, for new students joining the group. But I explain to them what happened. I'm always very open about it and say, this is to help, this is to protect you, this is to protect me. This is to make sure we do science the best way that we can and that we don't make any mistakes that could have been prevented. I think that, it's also taught me, it's made me re-examine other things like, should I be checking the sequencing that everyone does? Should I be going to that micromanaging level? And I do actually check all the sequencing of any new construct that's made. I do. I actually sign it off as it's being checked by Nicola. But I don't want to be that lab head that distrusts people first and they have to prove to me they were correct. I want to always be able to trust them first. So while we have lots of documenting raw data and putting it, kind of archiving it so that you've got the original file at the time so that there's repeated editing can't occur, or if it does occur, we can always go back to the original. I'm not going to be poring over everyone's raw counts to see if they've done anything sneaky. You have to trust people to some extent, I think, don't you? You can't. I don't want to be that person.
I think from my perspective, from the outsider's perspective, hearing about your story, the way you're telling it, I think it just quote unquote put you on a course that everyone should be on. I think the mechanisms that you put in place in your lab to make sure that the materials you're working with, the data is documented. It should be that way everywhere, all the time. And unfortunately, this is not taught. Some and basically I think that's how it should be everywhere. And I think I don't think it's gonna prevent you from changing your career or decreasing the length of your career. I think on the contrary, it's a noble thing to have come forward. It's phenomenal how you handled it. And I think it also got you to where you're supposed to be. Doing what you're supposed to be. I think as a person looking at it from the outside, you became the person you were always meant to be. And this was a hardship that had to happen.
Yeah. I'd like to think that I was always striving to do that. But I think that there was always that, how do we spin the story for the biggest impact as well? And that didn't mean that we were putting anything that was false. It didn't mean that we were doing anything dodgy. We weren't at all. But we were looking at the explanations for stuff.
Yeah.
And packaging the story in the way that was the most attractive and sexy. And that's what I know that people are doing. And I have little time for that now. And I think that it's going to take longer for me to publish now because I'm going to want to make sure that we have a really rock solid story. But I don't think that's a bad thing. And I think that the students that come out of the lab are going to have really good training. And I think it's not just that they're going to be trained well in all of that data management and stuff, but I think that they'll also know that I want them to know they're in a really supported environment and that, one of the things that when we were giving our talks that I mentioned was that we use electronic lab books and I give them a mock version. I get everyone to do a daily log so that I know what they're doing, so that I don't have to dive in and read every single page of their lab books. I need them to give me a high level of what they're doing. And when it's a critical point in their experiment, I then can go to that experiment and read everything that they've done. But I don't want to be checking that you split your cells today and going through their calculations on that day. I want to know when the Western blot's done and processed so that I can go through and check how they did the transfection and how they processed it and what were the expected molecular weights. So I can do that level of scrutiny. So these daily logs have been brilliant. And I actually brought them in before I went on maternity leave because how else do you keep running a lab while you're on maternity leave? And the team's always been fantastic about doing it. I'm constantly reminding them in lab meetings, keep up to date. I'll say, it's January now and you're still in December. And, I'm constantly telling them what month it is and, keep up to date. But in my example, I give them everyone gets given a template notebook to start with. I put in examples where they're reporting errors and mistakes. And that's because I want them to feel that it's a safe place to own your errors. I always say, you're going to struggle to make a mistake that's as stupid as the mistakes I've made. You know, where you've dropped something or you've put something in the wrong tube or, you know.
Or you use water.
Yeah.
You use water
yeah,
to do your agarose gel and then you wonder why your samples are diffusing.
Exactly, right? I don't care what they do. And I keep telling them, I don't care what the mistake is, so long as you've documented it and you've learned from it. That's, you know, we're not in a punitive environment. You're not going to be yelled at because you have done, if you've made a mistake. That's not what we're about. It's about being very open and working as a team. And my guys do work as a team. They are amazing people. I'm thankful every day to have each of them in my life. I just feel like every single one of them makes me a better person and a better scientist and just fulfilled just having those interactions with them. They're a really awesome group. We've got a team of 7 students at the moment and they support each other. They're great.
How do you select people to come to work in your lab? Because obviously these things sometimes when they're not organically, these communities don't form.
Yes, yeah. So I guess all it took was, the first student, and that was James Coleman, who was one of the affected authors for the science signalling. He was the first author. And he is just like I always say, James, you're my moral compass, you know, because he's always so kind and considerate of other people. And once I had him in the lab and I knew that he was such a great guy, I had someone to bounce off and vet people. And so the way that I recruit students now is that they talk to me, and I run my lab with a teaching academic at UNSW in Sydney. And so she and I will meet with them. And she's really important in the recruitment, actually, Angela Finch, because she teaches the students in undergraduate and they fall in love with GPCRs mainly because of her enthusiasm. And so they come to us, we interview them and have a chat, you know, tell them about us. We tell them to go and look at other labs, talk to everyone, and we tell them to talk to the current members of the lab, and if there are any past members of the lab, you know, that they should talk to them. And we also say, you know, think about how we're interacting with you now. And, you know, I'm very informal. I'm like, you call me Nick, never call me Dr. Smith, ever. And you don't even call me Nicola. You are part of my family. I'm Nick. You know, it's very different to how some people want to run their labs. I appreciate, but this is how I want to be with my team. We're all equals. After we speak to them, I then send them off with my students. Before COVID, they used to take them out for coffee. Mm-hmm. And the students then chat to them, and I always tell them, you're going to go and talk to my students, ask them anything. They're under instructions to be honest, and my understanding is that they are honest. And I always say that probably the first thing they're going to tell you is that Nicola swears a lot. And, you know, if you're uncomfortable with that, that's important that you know. If you're going to be uncomfortable with that, it's not going to be the right environment for you, because I've tried to stop and I put all my money in the swear jar and I just take it back out again. So they're very open as well. And the thing that I actually was speaking to one of the professors at uni, and she was saying, how do you always get such good students? And I was telling her this process, and I said that the students then tell me whether I should take the student or not. And she was appalled that I would give that, what she perceived as power, to my students. And that she goes, well, why would you let them choose? You know, I'm like, because they're in the lab with them all day, every day. And I trust my guys. I've got a great team. We don't have any bad eggs. We don't have any conflicts. They're all supportive of each other. And so they're the ones that are going to be the right ones to tell me if they've got the right feel for the person, or often they get other information out of them that's quite useful to know as well at the coffee. But I take that they end up having the ultimate say, and I think that that's what's maintained the really strong environment that we've got.
That's fantastic, and I love talking about these things because, as I mentioned on different occasions, not only on the podcast, but you want to read somebody's papers, you just go on PubMed, you read the papers, but talking to them about these processes, because unfortunately these things are not being taught during your postdoc, during your PhD, and these are so important. How to pick the people who would work for you, and how to manage everyday life, and develop processes in order to.
Sorry, I've been surprised since all the retraction stuff. I've actually had a couple of professors come to me and ask me for how I run my lab. They've asked for clones of my lab book. They've asked for, I have everything on OneNote as well, with like a little spiel of how we expect people to do things. And a big part of that is that they are to expect the best from me, and they are to expect that I will always do the best for them. In setting expectations, it's not a series of, you must record this, da, da, da. It's, I need you to record this so that I can stay on top of it. And if you feel I'm not staying on top of it, these are the ways that you are to bring me up to lift my game. So I've had people who've been researching, they've had their own labs for 20 years, and they're coming to me to ask for my processes so they can implement them. But I've also had a couple now of young investigators who have just been granted their own lab come and say, help me set up because I like the way you've done it. And those things are really important to me, that I'm able to help, because I had to learn the worst way what processes worked and what didn't. I love to be able to help, because we don't get that training. I'm terrible with budgets. I just spend the money until someone tells me I don't have any money. I don't. These are things I'm not equipped to do because I've never had any training in it. And you need strong mentors in all of these aspects. And people are just told, here, you've got a lab, you've got a startup. Good luck.
And you need to spend it, and then at the end, when you're ending up with the money, you're like, oh my God, what am I going to spend it on? And it has to go by this deadline, so you end up buying something that you may or may not use.
Yes, it's all of those things are really hard, and you need good mentors. And I've had some pretty good mentors in my life. I'm pretty lucky.
That's amazing. One of my questions about inspiring young scientists on the podcast is, what would be your advice to junior scientists on how to contribute to the field? But I think all our discussion so far has been about advice.
Yeah, exactly.
So, anything else you think you'd want to add?
I just think my advice would be, just do what you love, follow what you love, and work will never feel like work. There was a period in my life where work felt like work, and I didn't like it. And I often say, because after my I actually ended up at UNSW because I was told that people didn't think that my career would recover from having kids. And so I looked for a new role. And at that point, I really stopped and I had to reflect. And I actually felt like science had betrayed me, because I had loved it so much and I had given so much of myself to it. And then for that to happen, I just thought, I can't. Why am I doing this? Why am I flogging myself? Absolutely flogging myself for something that ultimately can't pay me back. And then I had to retract 2 papers. But now I say to my students who were there during all of that, I just say, thank you. Because of you, I've fallen back in love with science. And so I think that anytime you love what you're doing, it's not a job. And that would be my advice, is follow what you love. And I don't have any advice about trying to be a scientist with kids, because there's no easy way to do that.
It's hard. I think trying to have a career with kids, it's
Yeah, full
difficult.
stop. Yeah. Yeah.
But I think it's all about, I want to say all about prioritizing,
I think
making
that's
sometimes
reasonable.
difficult choices. And, you mentioned your then 3-year-old who said, no, Mommy, I need to send this email. And this happened
Yeah.
to me too, where
Yeah.
I was doing something and then my 4 and 5-year-old said, oh, you're working. You cannot play with me because you're working.
Yeah.
And that just
Yeah.
broke my heart.
Yeah.
And I
Yeah.
said, no, I'm not working. So I put down the computer and the phone and I went playing with him. But
Yeah.
yeah, those are difficult choices, but I don't think that should inhibit anyone from doing what they need to do in life.
Yeah. Yeah. I think the problem for us is that the granting bodies are trying very hard to make it clear that women who've had children or parents who have taken carers leave in whatever capacity have to be judged relative to opportunity. And right now, that doesn't take into account pregnancy or when you're back part-time, but you're still trying to juggle sick kids all the time. It's just taking that maternity leave or paternity leave into account, which is inadequate in itself. But when there's so little money, how does a grant review panel give money to someone who, had they not taken this time and worked part-time and had, I had 2 really bad pregnancies where I was on bed rest for 3 months and 4 months. I was wheelchair bound, in chronic pain. I mean, it was just, I do not, my husband always says, you don't do babies well. You don't do pregnancy well. And I always used to, before I ended up in the wheelchair, I'd be like hobbling around, and I'd always say to my grad students, don't have children. But none of that gets taken into account. But how do they then say, well, Nicola was on a really good trajectory. She does good science. We really like the way she thinks about orphan receptors. We think we should give her funding because this project's really cool. How do they choose that versus another project that's almost as cool or just as cool, but the researcher hasn't had to take that time out, and therefore they're able to say, I did publish these papers. It's not I think Nicola would have in that time had she been given the opportunity. They actually do have those papers. How do you, when you're looking at 8% success rates, give the funding to that parent or someone who's had a significant illness and had to apply for relative to opportunity? How do you give the funding to that person? And I think that's the biggest problem.
It is. It is. I wish we were math equations and you could get us all in the common denominator.
Yeah.
I'd be like, there you go. Now you're comparable.
Yeah, I know. Well, I wish we could be scientific about it. There's actually a petition going around in Australia at the moment because with the latest investigator grants that came out, so these are fellowships for the National Health and Medical Research Council, some of the kind of leading proponents of equality in research in Australia did some calculations, and they worked out that not only do we all know that there's that kind of cliff face that female researchers drop off when they hit a certain level, and then they just don't progress beyond that. And there are many reasons why that happens. But so there was less women in the higher levels, but for the women that did make it to the higher levels, they are systematically receiving less funding than the males. And again, many reasons for that, and probably because women are asking for less would be a big part of that.
Yeah.
But it just further underlines the fact that there's that systemic disadvantage. And I can't even imagine what it's like for a person of colour or, for someone from any kind of minority or marginalised group if a white, middle-class woman is facing those challenges when, you know, we have all of the privilege afforded to us other than being male. We're very, very privileged people, and still we're facing that disadvantage. I hope that this survey or this petition gains some more traction, and the funding bodies are trying to address it. It's just very, very hard to do so.
It is difficult, and I think it's, you know, you mentioned in the beginning, a couple of minutes ago when we were talking about, you know, embellishing a story in order to sell it. We all had to do it because that's how the system works, not because we wanted to. I always had a hard time, you know, writing fluff in the conclusion of a paper or in the abstract saying that we will cure something at some point with thanks to this discovery, because to me it doesn't mean anything. We've discovered something. This is important because it adds to the common knowledge about this class of proteins. But what will be the impact in 20 years? I have no idea. Will this discovery cure cancer? Probably not.
Exactly. You want to actually say this probably won't revolutionize medicine, but it's an important stepping stone in our knowledge. But you can't say that. In Australia, there's a huge, huge push for translational impact. So we need to now put our research all in terms of impact rather than output, but also we need to have a clear translational outcome. And there's so little appreciation for the importance of basic science in that, the fact that discovery science is discovery science. And I don't know where a discovery will take us, but if I haven't discovered it, no one will know where it could take us.
Exactly.
And there's an emeritus professor that I work with on a committee at my university, and we do gene technology regulation. And one of the things is you always have to say what the potential outcome would be of that genetically modified organism. And he makes people correct it to say it is hypothesized that this modification will cause this. And he said, it's my bugbear in science that you are not going about proving a hypothesis. You are testing a hypothesis. And in all of our grant funding agencies and in that concluding statement in our papers and our abstracts, we are attempting to prove that hypothesis rather than test that hypothesis by always saying that this work will lead to a better understanding of blood pressure control, and, you know, rather than saying we'll work out if this can lead to a better understanding. And in the back of my mind, like, what he says is absolutely right, and I want to try and communicate that better myself, but it's probably not going to fly. You still have to cure cancer every time you write a grant.
It's unfortunate, but unfortunately there is that need, because otherwise you won't get. It's fluff. To me, it's fluff. But it's something that we need to add. All right. So last question. And I asked this from everyone as well. Top 3 aha moments that you had as a scientist that molded your trajectory outside of the course corrections that we talked about scientifically.
I know that's such a hard question. I think for me, the aha moments in general are when something works, and you're in the dark room and you're waiting for that Western blot. Kids these days won't know what I'm talking about, but back in the old days when we used to actually have to develop X-ray film and something appearing. So one of my aha moments, that it didn't change the trajectory, but it just gave me that boost in the doldrums of your graduate, your PhD, and you think I can't do this anymore, was when I was making viruses, and I came in on Christmas Day to look after 72 T75s or T150s, trying to make a virus. And that moment where you look under the microscope and they're green, and you just think, oh yeah, I can do this. And God, this is cool. And you know that no one else would actually care about your green cells.
Yeah.
But God, that meant everything. Another time, I guess, would be when I was working with GPR 3701, and this was back when I was still in the lab doing it myself, and I'd made some constructs, and I was just made flip-in cells like I always do and ran them on a gel, and this receptor always appeared as two species. And I made a couple of truncation mutations, constructs, and I thought, hang on, I think this is protease, this is proteolytic processing of the N-terminus. So I just threw on some betimastat, which I'd used in my PhD for a different thing altogether. And I thought it was just one of those, let's just roll the dice and see what happens. And being in that dark room and going, this is a metalloprotease-mediated event. And then the next aha moment being when we got brain homogenates and showed that the balance was completely different in the brain where the receptor is highly expressed. So there's a different mechanism going on that you're getting just that cleaved version. Just the idea of how we could learn something about an orphan receptor with no ligands and yeah, I mean, it's not I haven't cured cancer, but it was just that moment of my hypothesis was correct, and it's meaningful because it's different in different parts of the body, and it has an impact on function of the receptor. That was pretty cool.
That's awesome. For me, it was always, not always, but I think I always love to do experiments in microplates, especially when it's on live cells, and then you come in on the day of the experiment and you look at your cells and they're beautiful and they're evenly distributed. And then you run your experiment and you're hurrying up to really quickly take a look at the data before you leave. You're not even saving the file, and then you plot it, and it's a beautiful sigmoidal dose response. You could have not picked better, you could have not picked triplicates because you don't have error bars, and then you're wondering, and then you repeat it a second time and a third time, and like, oh my God, biology and mathematics. How beautiful is that?
My I guess my third aha moment would be when I was in the Milligan lab and we started working with the first allosteric agonist for the FFA2, free fatty acid receptor 2. It's a short chain free fatty acid receptor. And the first time I tested that, both as an allosteric modulator and as an agonist, and I did exactly the same as you. I would always everyone used to laugh at me, I would always put my tubes on the counter as my negative control, and then I'd put my highest concentration so that I could stalk the counter and know when I went home at night whether that experiment worked or not. And then, when you're sitting there and you're plotting the data as it comes and you're watching those curves and those triplicates, and it's like, it's going down, it's real, it's real. It's an allosteric modulator, and God, that was exciting. And then I just pumped out data because I was so excited. I was setting up stupid, ambitious experiments, which I churned through and just had bins and bins of radiation waste coming through, but just buoyed by the excitement of that data as it came off and plotting it. And love it.
Yeah. And sometimes it happened that it was very late at night, no one was around, and you were the only one dancing around in the lab saying, it finally worked.
I know. Woo-hoo! You think, well, no one else cares. I'll go home and tell the cat. Yeah.
Exactly. All right. Thank you so much, Nicola. I think it was fantastic talking to you. Thank you so much for coming on board, telling your story. I can't wait for this episode to be released.
Thank you for the opportunity. I've really enjoyed the chat and I love the series. So thank you very much.
Thank you. Talk to you soon. Thank you for joining me and listening to the Dr. GPCR podcast. I'd like to thank Dr. Nicola Smith for telling her story and sharing it with me and you. Also, a big thank you to our team members, Attila Forrest and Ines Pinero. Become a beta tester of the brand new Dr. GPCR Ecosystem 2.0 today by visiting ecosystem.drgpcr.com. Also, we invite you to subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.